This discussion is about the molecular basis of inheritance, which of course is about DNA and chromosomes, from Chapter 16 of Chemical Biology. So, as we know, early in the 20th century, figuring out what molecule was responsible for inheritance was a big challenge for biologists. Thomas Hunt Morgan and his group had shown that the genes, the heritable units, were located on chromosomes, but chromosomes were made of, as they knew by that point, DNA and histones or other kinds of proteins. And so they didn't really know which part of the chromosome was responsible for the inherited units. So they had to figure out animals they could use to study with and so forth. And so first they used bacteria and viruses to study a lot of this stuff. The learning that about the role of DNA was first begun with the research by Frederick Griffith in the late 1920s. He worked with two different strains of a bacterium, one of which was pathogenic and one was not pathogenic. And then when he mixed heat-killed pathogenic bacteria with the living non-pathogenic bacteria, he found that when he added those bacteria to, or when he injected them into living animals, it caused the pathogenic, they had become pathogenic in the process. This shows the different kinds of cells that he had here. This is the one that caused the death of the mouse, and this one did not. And so he killed these, in which case it doesn't hurt the mouse. But when he added those together with the living non-pathogenic ones, the mouse died. So something in the dead cells transformed or changed the non-pathogenic bacteria to pathogenic ones, but it still remained to discover what molecule that was. Most scientists at the time thought proteins, because there were so many different kinds of proteins, and DNA just seemed to be rather static, and it was just a basic molecule, it seemed like, although very big. In the 1940s, then Avery and McLeod and McCarty figured out that the transforming substance was DNA, and they did this by tagging DNA in the proteins with different substances. More evidence came from various kinds of things, and so when you looked at what happened with the bacteriophages, which inject their DNA into the cells, you could tag the outside with one isotope in the DNA with another isotope and see which one caused the problem. And so that's what happened in 1952. Alfred Hershey and Martha Chase performed experiments using a bacteriophage, a T2 bacteriophage, and they tagged the proteins with radioactive sulfur and the DNA with radioactive phosphorus. And so they were testing this two different batches here to determine which one caused the change in the bacteria. So what they found was that the phage DNA that was injected in there is what caused the problem, and the protein shell was on the outside. And so then when they looked at it, then what they saw was the radioactivity remained in the liquid, but the pellet with the bacterial contents did not have the radioactivity. And when they did the same process to the second batch with the radioactive phosphorus, then the radioactive DNA was in the pellet, so that indicated it was a DNA that was the transforming substance. Chargaff in 1950 showed that the percentage of adenine was always matched the percentage of thymine, and cytosine always matched guanine. And so this, as we now call the Chargaff's rule of base pairing, this gave a little bit more credence to understanding what DNA was doing. But the understanding of the construction of the molecule was something that eluded them for a little bit longer. There were a number of scientists working on this. Okay, they knew that DNA was made of these nucleotides, and they knew the nucleotides were sugars, deoxyribose sugar, and one of the 4 nitrogen bases and a phosphate, but they didn't know how they were put together. And so more scientists were continuing to work on this. Wilkins and Franklin were using X-ray crystallography to study the structure. Rosalind Franklin made a picture of DNA in the crystalline, the X-ray diffraction picture that looked like this. Earlier, as we've seen, Francis Crick had shown that this is what a helix would look like in an X-ray diffraction picture. And so this was something that was one of the final keys to understanding how this worked. Okay. it showed or suggested that DNA was made up of two strands that formed a double helix. And so the chemical structure was beginning to become a little bit more clear. Watson and Crick took the ideas that they had gained from looking at the evidence that other scientists had learned and collected, and figured that these backbones, the sugar phosphate backbones, were antiparallel, running in opposite directions, and that the bases were paired like with a purine with a pyrimidine. And this gave a consistent X-ray picture like you saw. If they put purine with purine, then some portions of the molecule would have been wider, and pyrimidine with pyrimidine, some would have been narrower, so you would have ended up with a back and forth motion. Whereas when putting a purine with a pyrimidine, as we see with adenine with thymine and guanine with cytosine, then you're going to have consistent width of the molecule, which is what you see in the X-ray data that Rosalind Franklin gathered. So they figured this was more specific, and they looked at what Chargaff had learned, and along with the things that Franklin and others had determined, and they built a model to show how DNA was constructed. And this was kind of the beginning of our understanding of how DNA works. So we have our bases, our 4 nitrogen bases, adenine and thymine, pair together with 2 hydrogen bonds here. Guanine and cytosine pair together with 3 hydrogen bonds. And they were looking at maybe that's the way that this could be copied, because since there's specific base pairing, maybe there would be a way that this worked together. The strands are complementary to each other, and that means that each strand serves as a template for making a new strand in replication. So the next problem was to figure out how the molecule replicated itself. So here we have our current understanding of how this works. We have a parent molecule here. We have enzymes basically that in heat that separate the strands, and then we have enzymes that can plug the correct nucleotides into place, making 2 daughter molecules. And now we understand that each one is consisting of 1 original strand, parental strand, and 1 new strand. How to show that that was really what happened. So Watson and Crick assumed or thought that it was probably this semi-conservative method that we talked about there. Other people were saying no. They thought maybe it was conservative, that the 2 parent strands would open up and make copies and then zip back together, and the new strand would be totally new. And then there were others that thought, well, the whole thing seemed to be might be chopped up, and each strand was a mixture of old and new. And this just shows what the different ideas were that people had. So how to go about showing which one was correct. There were experiments by Meselson and Stahl that supported this semi-conservative molecule. So what they did is they labeled nucleotides with the old strands with a heavy isotope of nitrogen, and new nucleotides were labeled with a lighter isotope. And so what you get with the first replication is you get a hybrid band. Okay. So here's what you saw with these different things, and the DNA was centrifuged here. So if it had been conservative, then you would end up with a heavy band at the bottom and lighter bands at the top, because the newer bands would be made with the lighter isotopes. The dispersive model, you would see no real separation of bands. You'd see they're all intermixed with each other, and the semi-conservative would show a heavier band and a lighter band. And this is what you saw eventually after they did the experiment to see what happened. DNA replication is very quick in terms of how fast it happens, something on the order of, I think, 500 nucleotides per second or something like that. There are a number of different enzymes that are involved in the replication process. It begins, first of all, at sites called origins of replication, at which places the DNA strands are separated, opening up what we call a replication bubble. And then replication is going to proceed in both directions from the origin until the entire molecule is copied. In bacteria, there is one origin of replication. Since they have 1 chromosome and it's circular in shape, they have 1 origin of replication, and then the replication proceeds from there. 1 bubble with 2 replication forks, 1 on each side of that, and that produces our 2 DNA molecules. And a eukaryote, since there's so much more DNA and the DNA is linear rather than circular, Then you may have multiple origins of replication. So we have multiple bubbles, and over time we're going to get to daughter DNA molecules. And so you see the micrographs of the bacterial genome and the eukaryotic genome showing basically what we're showing in the diagrams here. At the end of the replication bubble, those forks are the areas where the DNA strands are elongating with each other. And there are numerous enzymes involved in this process. Helicase is an enzyme that unzips the strands or untwists it. Topoisomerases correct the overwinding. When you pull two strands apart that are wound around each other, they end up getting tighter at the end away from where you're pulling. And topoisomerases are enzymes that actually cut or break the DNA and swivel it around and then rejoin it together so that you don't end up with that overwinding occurring. So here's the topoisomerase working ahead of the replication fork. Helicase is moving this way to unzip. We have our primase adding the RNA primers, and we have binding proteins that kind of hold the strands separate when they're in single strands. DNA polymerases are molecules that are going to add the appropriate nucleotides, the matching complementary nucleotides to the template strands. And they can't start the synthesis. They have to add nucleotides to the 3' end of the molecule. And so what they start with is actually a short RNA primer, which is really interesting. It's one of the things that leads most scientists to believe that RNA came before DNA, and then DNA was derived from RNA. Primase can start the RNA chain from scratch, and it adds RNA nucleotides one at a time. The short primers are added at the 3' end, and that allows for the addition of the nucleotides in sequence there. DNA polymerases catalyze this elongation, and then they require a primer and the DNA template strand most of the time. About 500 nucleotides per second in prokaryotes and about 50 nucleotides per second in human cells. Still very fast. The nucleotides that are added are called nucleoside triphosphates, kind of like adenosine triphosphate is an RNA nucleotide with a couple of extra phosphates on it. So the sugars are different. The DATP has deoxyribose instead of ATP having ribose. And so each one of those are going to join the strand. It's going to lose 2 of those phosphate groups as a molecule of pyrophosphate. So we can see here what happens. Here's our nucleoside triphosphate, and this side it's thymine triphosphate, or D-thymine triphosphate, and 2 of those phosphates are going to break off in the process of adding the thymine onto the growing chain there. The antiparallel structure is what's going to affect replication. Basically, one side gets to copy continuously, the other side has to copy in short stretches, because the DNA polymerases can only add nucleotides to the free 3' end and not the 5' end, so the new DNA strand can elongate only from 5' to 3' as it's elongating. So one strand of DNA that can copy continuously is called the leading strand. The other strand that has to copy a piece at a time is called the lagging strand. So here's our leading strand and our lagging strand. The leading strand, again, from the origin, can copy continuously there, whereas the lagging strand can only copy a little bit at a time because it has to copy backwards. And so actually the machinery of all the transcription factors are going to pull part of it out and let it copy backwards and pull out another part, and so forth. The lagging strand is made of a series of segments called Okazaki fragments, and they're going to be joined together by DNA ligase, which kind of tapes or glues or staples the fragments together. So here's our origin of a replication, our leading strand and our lagging strand. And on this one, this is the leading strand, and this is the lagging strand. And so we're going replication in 2 directions at the same time. Here's our template strand with the primer being added by primase. There's our RNA primer for fragment 1. Then we have our Okazaki fragment that's forming by means of the DNA polymerase. And then we have another RNA primer and our second Okazaki fragment. And then the primase is going to remove the primers and add DNA nucleotides and end up with the ligase producing our entire fragment, or gluing our entire fragment together. This shows all of the different kinds of molecules that are involved in this. Couple of different DNA polymerases and primase, helicase, of course, topoisomerases, DNA ligases, and so forth that are involved in all of these complex reactions. The proteins that work in this process are part of what we call a DNA replication machine, and it can be stationary, but there's also a lot of motion going on in the process. The DNA seems to be pulled through and reeled out basically through the whole process. The video that we watched in class from the DNA LC or DNA I site showed how that worked, and you can find that video. I'll link it on our Edmodo group. This kind of shows you the process. So we've got one side, the leading strand is being copied continuously here, and the lagging strand is being pulled out, copied part at a time, and then another part gets pulled out and copied, and so forth. Now, DNA polymerases are also involved in proofreading the DNA to repair incorrectly placed nucleotides. They're going to correct those errors in base pairing most of the time. There are damages or mistakes that sometimes do not get caught. So there are things like cigarette smoke and x-rays and various other kinds of mutagenic agents that can cause these mistakes to occur. Sometimes the mistakes are just mistakes, they're just spontaneous changes, but you have basically a nuclease enzyme that cuts out the damaged stretch of DNA and repairs it. So here's one that we have a mismatch of our bases here, and so the nuclease is going to cut those out. The DNA polymerase is going to come behind and fill in with the correct nucleotides, and then ligase is going to glue them back together. Now, there's not a zero error rate. There are still going to be some differences there, and so sometimes you're going to have sequence changes that get passed on. And these are mutations that end up as the source of genetic variation. Sometimes the variations are good, sometimes they're bad, sometimes they're neutral. They're just differences, but there are differences in the genome from one person to the next. When you get to the end of a DNA molecule, it's kind of hard to continue because that requirement to copy from 5' to 3', it's going to have not have a way to complete the 5' end. And so what you end up with is shorter and shorter DNA molecules with the ends being uneven over a period of time. For prokaryotes, this isn't a problem because they have a circular chromosome, so there's no end to the circular chromosome, but in linear chromosomes, it gets to be a problem. And so you can see here, when that RNA primer is removed from the 5' end, then there's no way for the DNA polymerase to come in and add these nucleotides to this particular part. So over time, the chromosomes will get shorter and shorter after multiple rounds of replication. In eukaryotes, there are special sequences on the ends of chromosomes called telomeres. The telo part means the end, okay? And the telomeres don't prevent the shortening, but they postpone the breaking away of genes that are located near the ends. And so some people think that the shortening of telomeres is related to aging. It's a good question. I don't think anybody knows. This picture shows some chromosomes, and it shows the telomere sequences that are highlighted there on the ends of the chromosomes. So you can see where they are. There is an enzyme called telomerase that can catalyze the lengthening of telomeres. This occurs mostly in germ cells. Those are the cells that produce the gametes. And so, because those would definitely be a problem for the gametes that they produce. And so, this could prevent cells from becoming cancerous by limiting the number of cell divisions. However, some cancer cells have evidence of telomerase activity, so that may be what allows cancer cells to continue dividing as often as they do. There are some cancer cells that activate or produce their own telomerase. Chromosomes are made of DNA packed together with proteins. The DNA is coiled and then supercoiled together. There are a lot of proteins involved here. There are various different ones. Chromatin is what we find in the nucleus of eukaryotic cells, and the chromosomes are definitely packed very tightly into the nucleus there. So here's our double helix. Wound around some proteins called histones. Usually there are packets of histones, about 8 histones there in a packet with the DNA wrapped around a couple of times, and then those kind of start coiling around each other. So we end up with our nucleosomes here, and then we have our beads on a string. We have a 30 nanometer wide fiber here of all those nucleosomes packed together, and then that starts to loop back on itself. There's the loop domains here that's about 300 nanometers in diameter. And then our replicated chromosomes here are about 1400 nanometers. So we end up with a looping and a twisting and a curling back on itself over a great distance, shortening that DNA molecule that's about 3 feet long in each one of your cells into a small, tightly packed structure that can fit, multiple of them fit inside the nucleus of the cell. The chromatin goes through all these changes. Again, we've got a 10 nanometer fiber, 30 nanometer fiber. That's due to the folding and looping. The measurements, I think, aren't really that important as long as you understand that there's looping and twisting and curling around these proteins that are involved. Most of the time in the nucleus, during interphase, then the chromatin is loosely packed together. We've talked about that before. That's called euchromatin. In interphase of the cell cycle, there are a couple of places in there that are highly condensed into areas called heterochromatin. That's going to be like the centromeres and telomeres are going to be tightly condensed there. That makes it difficult to express genetic information in those regions. As you can imagine, it's kind of tightly wound up there together. The histones themselves can be chemically modified, and that can result in some changes in the organization of the chromatin. And so we'll talk more about what happens next with our next unit, next chapter.